Good morning, everyone. So we have started this embryology sessions of the GI system development. We are going through the gross anatomy, practical anatomy of GI tract, and the systemic embryology that needs some kind of like a media support, like it needs some animation, some short clip videos to understand the process, what is going on as we are going through the development of the GI tract. Now, we are going to explore to this general features of this particular GI system development. Whenever we are going to talk about this embryological development of the GI tract, we break it down into three segments. Like we talk it like a foregut, midgut and hindgut. So, we break this particular entire GI tube into three segments. Foregut, that means the foremost part of the GI tract, the middle portions, and then the last portions which deals with this particular sigmoid colon, rectum, anal canal, etc., etc. And each particular part has a different or kind of like a unique way of developing and transforming into different parts of this foregut. And while we are going through this particular GI system development, we are going to encounter a peritoneal folds because it is intensely wrapped around this particular GI tube which is present to it. Along with that, in the GI tract, we have number of organs which are present. So, that organ also develops as this particular GI tube evolves, develops, matures. So, this development of these particular organs related to this hepatobiliary apparatus, pancreas, etc., will be coming in. Now, this particular spleen, which is a part of GI tract placed in here, is not in a true sense or not belongs to the true family of the GI system. It is developing from this particular reticuloendothelial system tract. That means, it is coming from elsewhere, placed in the GI tract, stays in there, and as it is living in this particular GI system, we talk about the development of the spleen along with this GI system organ development. Now, once we have understood this particular element, we are going to talk about some of the developmental errors which are materially important to us, clinically important to us, and as well as we are going to see the clinical cases in our clinical life. So, without waiting much, we are entering to this developmental part of the GI system development. Now, this GI tract, as you see that it begins with this oral cavity and it goes down from the oral cavity to the pharynx and then goes down to this particular tube, we call them as esophagus. That esophagus ends into the stomach, and then we have this particular coils of the small intestine starting with this duodenum, jejunum, ileum, and we have the cecum appendix which is the beginning of the large intestine. Then we have the ascending colon, then we have a bend, then we have the transverse colon, then again we have a bend, then it is dropping down, we call them as descending colon, and the sigmoid colon, and then rectum and anal canal. This is how this entire tube system is developed. So, we will be emphasizing on the development of this particular esophagus in here and beyond esophagus in the gut tube development. Along with that, we are going to talk about the liver, gallbladder, pancreas and spleen as well, which is developing in this particular segment. So, we have 4 or around classes to talk about this particular GI system development, see how we can cover into this particular developmental detail which is required for us. Now, histologically, if we are going to bring it down into micro sections, we already have seen this gut or tube have got 4 layers. Like starting from inside outwards, we have the mucosa, then we have submucosa, then we have the muscle layer which is divided into 2 things, outer longitudinal, inner circular, and then we have the serous layer which is outer binding coat. So, we have 4 layers which is present histologically. Now, if you break it down, the mucosa, it has got sub layers in the mucosa. It has got cells along with the glands, we call them as epithelium with the glands. Then it is resting on something, we call them as lamina propria, this is a basement membrane. Then under to it we have a muscle layer, thin muscle layer which moves along these particular coats. So, this muscularis mucosae. So, we have this particular 4 coats and 1, 2, 3 coats present in here. Now, if you just like break it down, this one layer which is epithelium and the glands source from a different source. And rest of this particular thing, which is lamina propria, muscularis mucosae, submucosa, muscularis externa, serosa, all these layers are sourced from elsewhere. So, the only thing which has a different origin, which is this cells and the glands present in here. So, these cells and these glands develops in or develops from a part we call them as endoderm tube, also known as primitive gut tube. So, this particular thing which is known as endoderm or gut tube, which is giving rise to the cells and along with the glands, and else things, whatever things are you have charted in here, is developing from the mesoderms. So, this particular mesoderm can be divided in a splanchnic or splanchnoporheic, or which is coming from the lateral plate mesoderm. So, splanchnic mesoderm is which is coming from the lateral plate mesoderm, which is going to yield the rest of the structure. So, this is what we are starting with, or the starting point of the GI system development is that gut tube. So, all the gut tube that we are going to talk over here, the prime thing about this particular GI system development, we are going to talk about the gut tube, where this gut is coming from and how this particular gut is developed. So, this particular gut tube develops from a primitive or old tube, we call them as primitive gut tube. So, where from this particular primitive gut tube is formed, that is what we are beginning with. So, we call it formations of the primitive gut tube, how this tube has get into our system and develops into this different parts of foregut, midgut and hindgut. So, this is what we are starting with, or formations of the primitive gut tube. So, this is a like a embryo which is in a phase of development, likely in this like 4th week or nearly 4 week, 3rd week of development, and this particular 3rd week of development embryo with time evolves into segments. So, this is a bilaminar germ disc which has become trilaminar germ disc, and it has 2 cavities on here. Above we call them as amniotic cavity, and bottom one, the here bigger one, we call them as yolk sac or yolk sac cavity. These are the 2 things are there, and these are the germ layers. That means ectoderm, mesoderm, endoderm, they are placed in this particular layer. So, this is where this embryo is going to begin or begin with. So, this area we call them as stomodium end or proctodium end, which is on the other side. So, this is the future mouth side, and this is the future tail side or anus side, which is going to hide. Now, you see that this is the early phase, this is phase little bit more advanced, and then we have the further advanced thing. So, what happens with the times, we have a phenomenon we call them as folding of embryo. That means, this particular tube starts to have unrolled in or have to develop into something a bend. We call them as folding on the cranial side and folding on the caudal side. So, we call them a head fold and tail fold yet to begin, and with this arrow which is having 2 points over here, we have this particular embryo starting to bend down. We call them as folding of embryo on the cranial side and folding of the embryo on the caudal side. Now, afterwards what happens, we have a lateral fold or lateral bend starts in. So, as this particular lateral bend folds in, this particular yolk sac slowly gets into this particular germ disc, and gradually this particular shape takes the shape of this particular tube and slowly gets absorbed into this. So, this yellow thing which is the yolk sac slowly getting into the developing embryo, rolled in like a tube from the flat page through this particular process of head fold, tail fold and lateral fold. So, it is gradually assume it is like a paper, slowly it is rolling down and taking a shape of tube, and as this particular folding keeps on happening, this tube thing slowly getting incorporated or absorbed into this developing embryo. So, we have a other visual on this particular developmental folds like this. So, this is a yolk sac which you call them as endoderm, which is present on here. Slowly the yolk sac with this particular folding slowly getting absorbed within this developing embryo and takes a shape of a tube like this. And this particular yolk sac makes a tube like this and makes a thin slit connections, and the remaining of this particular yolk sac which becomes like this. So, eventual later, like after 4 weeks of age, this particular yolk sac becomes this small. It makes a kind of tube, rest of the things we call them as endodermal tube, and a connection between the tube which is present in the embryo, we call them as primitive gut tube, which becomes endodermal tube here, with this yolk sac tube has a communication channel, we call them as Vitello-intestinal duct. So this whole thing, yolk sac, basically pumped in, slowly get into this particular developing embryo and forms a tube inside. That particular tube we call them as primitive gut tube. Now this particular primitive gut tube which is present in this particular head fold region, we call them as foregut. The primitive gut tube which is present on the tail fold region, we call them as hindgut. And the middle portion which has a communication with this Vitello-intestinal duct, we call them as midgut portion. So this is how this can be separated, this is how. So the gist of the thing is that so far whatever we have talked over these 10 minutes is that where from this primitive gut tube is coming from, that's one word, yolk sac tube or yolk sac, which gradually becomes a sac into tube, and that particular tube has a communication with the remains of the sac which becomes like this small tiny like this, and these connections we call them as Vitello-intestinal duct. Now this Vitello-intestinal duct should normally get absorbed, completely should get vanished by end of 5 weeks. If it fails to vanish, we have a disease. That's what we are going to talk about. So out here so far what we have talked, we have one line of things we have acquired knowledge on. The gut where is coming from, or the tube which is present in these intestines, which is called them as gut tube, which is coming from the primitive gut tube. So this where from primitive gut tube is coming, is from yolk sac. The yolk sac, what happens to it? It was there as a sac, it becomes a tube, and that particular tube gets inside the developing embryo by this particular folding process which happens during this particular process of the embryonic development. What happens to the yolk sac? It becomes a very thin tiny remnant of the sacs, we call them as umbilical vesicle like this, and this tube remains with the communication which is known as Vitello-intestinal duct. That should normally also get vanished by the end of 5 weeks. So this is the fate of this particular yolk sac, and that particular contributes to form the gut tube. So that particular gut tube present in here yields the entire intestine, or yields this particular esophagus, yields this particular stomach, small intestine, large intestine, everything else. So this is what we have to understand. Now the ends of this developing embryo, this is the foregut end that we call them as this cranial end, and this is the tail end. Now out here within this particular mesoderm or this particular mesenchymal element, we have a depression. So this mesoderm over here condenses to form the future heart tube. You can see that in the picture B, it starts to form a spherical structure. This particular spherical structure is the formation which is going on, the future heart which is going on the verge of the head fold region. So as this particular head fold keeps on happening, the brain starts to develop from here. So this particular brain or neural tube, as it starts to form over here, it bulges over. As it bulges over, the gut and along with this particular heart slowly gets put inside, pressured inside to it, and slowly the heart tube which has developed from the neck region or future neck region goes down to this thorax region because of the developing brain which bulges on here and pushes the heart and this particular developing region over here. Now what is point of concern regard to this particular GI system development? This bend where we have a part of this particular gut tube invaginated in here has a depression point. So this depression point separates from this particular future mouth region, we call them as stomodium. And likewise in this particular tail region, we have this particular cloacal region which is present in here, we call them as proctodium. In between this particular gut tube and this depression, we have a membrane which is present in here, we call them as Buccopharyngeal membrane. That particular membrane is separating the tube from the particular mouth. That particular mouth which has to be communicated through the gut tube if this particular buccopharyngeal membrane ruptures. Similar to it, at this particular caudal end where this particular cloacal end is there, where we have this particular late part or hind gut tube is there, there is a depression. That depression we call them as proctodium. The proctodium is separated from the hind gut tube by a membrane, we call them as cloacal membrane. That particular membrane has to rupture to give rise to this particular connection communication. By any chance, if this membrane fails to rupture, we have a tube, but that particular tube is not communicated through the mouth, or that particular tube is not communicated through the anus or anal canal. So, then we have a clinical conditions. Again, we will be coming back when we going to talk about the clinical scenarios or going to discuss about this clinical problems or developmental error. We will be coming to this particular cloacal membrane. We will be coming to talk about the buccopharyngeal membrane again and again, and we will be referring to the stomodium and proctodium. So, you have to keep this word in mind, stomodium and proctodium and buccopharyngeal membrane and cloacal membrane. So, we have learned so far few things regard to this particular embryonic development. So, not to make this particular thing communicated or complicated by any ways, we learn this particular thing, we call them as Yolk Sac and fate of Yolk Sac, which goes to form this primitive gut tube, and that particular primitive gut tube forms this gut or gut tube, which is present in our body or which is present in the system that we have. That is what we have to keep in mind. The Yolk sac becomes a vesicle and it forms the primitive gut tube. The vesicle part normally should get absorbed. That vesicle part is communicated later on with the duct channel. We call them as Vitello-intestinal duct. In short, we call them as VID. That particular VID also should get completely vanished. If it fails to vanish, then we have a clinical case or clinical situation. That is what we are going to talk about later on. So, this is about the thing that you have to know. Now, as this particular tube is present or going to form, it has a depression in the cranial end or caudal end. We call this depression as stomodium and proctodium. So, this particular stomodium and proctodium having a membrane present in here. We call them as Buccopharyngeal membrane and cloacal membrane. This membrane ruptures, then we get this particular anal canal communicated with this anus, then the fecal matter can be going out, and this particular stomodium ruptures with the buccopharyngeal membrane ruptures. So, this particular mouth cavity communicates with this particular gut tube. So, we have this mouth cavity and anal cavity which is communicated through the anus and this mouth through the rupture of this particular membrane. So, stomodium, proctodium, buccopharyngeal membrane, cloacal membrane, the significance of it, and the Yolk sac, the fate of Yolk sac, and remnant of the Yolk sac which is communicating over here as Vitello-intestinal duct, and that particular Vitello-intestinal duct is normally should have absorbed completely. Rest of the things, if it is failed to absorb, we will deal with these things, clinical problem later on. Other than this, in this particular hindgut tube, you see that there might be another projections. We call them as diverticulum. This diverticulum, we call them as allantoic diverticulum. We will talk about the allantoic diverticulum again when you are going to talk about the urinary system development. That is separate. So, I am not just addressing that part. So, we are only focusing on Vitello-intestinal duct, Yolk sac, primitive foregut tube, primitive midgut tube, primitive hindgut tube. That is all. So, with this, we are moving forward. Now, we are getting a tube which can be divided into three parts: foregut tube, midgut tube, hindgut tube. We understand which is foregut, which is midgut and hindgut. Easy to understand the midgut, which is communicated with this primitive Yolk sac through this channel. We call them as Vitello-intestinal duct channel. Rest is in front of that we will be having foregut, in the behind to it we have a hindgut tube. How these things have formed? Because of this folding which is happening cranially, caudally and laterally, making this particular tube to have formed inside this developing embryo. So, each of this particular foregut, midgut and hindgut which you have formed, we are going to talk about how this particular foregut tube transforms into what are the structures. Midgut tube gives rise what are the development adult structures, so is the hindgut tube. That is how we are going to talk about. So, this is about the thing which is talking about. Now, do not forget this particular buccopharyngeal membrane, cloacal membrane, proctodium, stomodium, etc., because these are MCQs which commonly surfaces during your exam questions. Now, taking from the conversions on this particular tube. As this particular tube is formed, this particular tube is wrapped by a double layer membrane, we call them as mesentery. Now, this particular mesentery wraps on this particular primitive gut tube and hangs this particular gut tube in here. So, this particular mesentery can be divided into two parts in embryologically, how we are seeing it, and this developmental and transformations of the mesentery is important and easy to understand. You see that this is the gut tube which is formed here, and then this particular gut tube is wrapped by this particular membrane like structures which is present in here, we call them as mesentery or peritoneum fold. So, this double layer membrane, we call them as mesentery, can be divided into two parts. This is the posterior wall of the body, this is the anterior side of the body. So, this mesentery have got two things which is present in here. It is hanging from the posterior wall and ending in this particular gut tube, and it is hanging from this anterior body wall and ending into this particular gut tube. So, it has two segments, say for example, segment over here and segment over here. So, this is the back side, we call this mesentery as dorsal mesentery because it is attaching on the dorsal body wall. This mesentery which is present in the front side of this gut tube, we call them as ventral mesentery. So, mesentery embryologically we divide into two parts, ventral and dorsal. In simple words, as they are attached to these different body walls, posterior body wall, if they are attached to, we call them as ventral mesentery. If it is the dorsal mesentery. If they are attached to the anterior body wall, we call them as the ventral mesentery. So, we have this particular mesentery placed in here, dorsal mesentery and the ventral mesentery. Now, this ventral mesentery is short, the dorsal mesentery is long, present throughout this particular length of this particular gut tube. So, this ventral mesentery exists in small parts of this particular gut tube. This is present in only the foregut tube or parts of the foregut tube only. The middle portion, the hind portion, this particular ventral mesentery do not exist. So, this particular foregut tube only have this particular ventral mesentery. Midgut, hindgut do not have it. So, this particular transformations of the mesentery that we have to know, and because they are our questions, we have to understand this particular mesentery transformation. Now, you see that this particular picture we are focusing on. So, this is the gut tube, primitive gut tube which has taken some shape over here. So, we will be focusing in this particular picture. So, this particular area is anterior body wall which is yet to develop fully, and this is posterior body wall which is have developed completely more or less. Now, this is yellow one which you seeing here is a gut tube and little bit progressed, little bit have taken shape. Now, this particular gray shadows which is present in front of this tube and the back of this, we call them as mesentery. Now, in mesentery, this part we call them as ventral mesentery which is attached to this anterior body wall. This mesentery which is present on the back side, we call them as dorsal mesentery. Now, you focus on the dorsal mesentery, they are present on entire length of it. So, this dorsal mesentery which is present on entire length of it have got different names. So, this dorsal mesentery which is present on the back of the stomach, we call them as dorsal mesogastrium. The dorsal mesentery which is related on the back side of duodenum, we call them as mesoduodenum. Back side of the jejunum ileum, which is the mesentery proper that you see when you are going to take out this particular gut tube, we are going to see this mesentery which is we call them as the mesentery which is wrapping around the jejunum and ileum. Colon around that we have this mesentery, we call them as dorsal mesocolon. So, embryologically this particular element we call them as mesentery, posteriorly they are present in entire length, have got different names. Now, coming to the front side, we have only this foregut, only this particular region have got mesentery, we call them as ventral mesentery. So, only in the foregut tube region we have this particular ventral mesentery, and rest of these things do not have it. So, this is a different thing which is present in terms of the mesentery, ventral mesentery and dorsal mesentery. Now, out here we focus in this particular ventral mesentery first, because the dorsal mesentery we are going to see that later on, we will be dealing with later on. The small one we will deal first. So, this particular narrow strip of mesentery that is present in the ventral body wall and ending in the tube has got remarkable thing. Now, out here we see that this tube, what this tube looks like? It's a stomach, right? And in front of the stomach we have this particular anterior body wall which is formed. So, upper portions of the anterior body wall we see that this is developing, and this is the future umbilical region. Lower part haven't formed yet, because this is how this particular body wall, anterior body wall develops. Now this particular mesentery we call them as ventral mesentery. Now what is missing out here in front of the stomach? Is there anything missing in between the anterior body wall and the stomach? Liver. So liver has yet to come. So we place the liver in between them. Now if you place the liver in between in front of the stomach, it splits this particular ventral mesentery into two parts. Part anterior to the stomach, part anterior to the liver. Isn't that? So this particular ventral mesentery, this one, and between the anterior body wall and the liver, this ventral mesentery takes a different name. We call them as falciform ligament, that we have seen that attached on the margins of the liver. So this liver placed in develops over here in this region, splits this ventral mesentery and takes this part of the ventral mesentery within these developing areas and makes this particular ventral mesentery within this particular ligament like structure, that is we call them as falciform ligament. Now what happens to the posterior to the liver, between the liver and this particular stomach? We have this narrow strip of mesentery present. This narrow piece of mesentery over here, we call them as lesser omentum. Now what happens to the greater omentum? The greater omentum is this one, which is present as on the posterior side of this particular greater curvature of the stomach, which is a part of dorsal mesentery. So this transformation of this particular ventral mesentery, which is transformed because of the development of the liver within it. It splits this ventral mesentery into two parts. A part develops in front of this particular anterior body wall to the liver, becomes this falciform ligament, and the part posterior to liver and in front of the stomach becomes a part of mesentery. We call them as lesser omentum. So this is how the lesser omentum and the greater omentum in related to the stomach has happened, and this is how this falciform ligament has formed. These are all your MCQ questions. So how this particular one, which embryological structure gave rise to the falciform ligament, which embryological structure give rise to this lesser omentum and greater omentum likewise. So ventral mesentery, dorsal mesentery. Again, to make it simple, if you have lost in between, so three things you have to focus. Mesentery are of two types. What are the two types? Ventral one, dorsal one. Ventral one entirely present? No, it is partly present within only the foregut region. Dorsal one is present throughout the length. Now take the small one first. The ventral one, which is present in this foregut region, is present in between the anterior body wall and the stomach. Now with the development of the liver, which placed in between the stomach and this anterior body wall, splits this particular ventral mesentery into two parts. Anterior to it, falciform ligament, posterior to it, we call them as lesser omentum. The part which are left with other omentum, which we call them as greater omentum, who transform it. This particular greater omentum is formed from this dorsal mesogastrium, the dorsal mesogastrium which is present on the posterior side. So this transformation is again very important for us for the MCQ reasons, and this liver, how this particular falciform ligament is there, we have to know. Now if you address it over here, you see that the extent of the falciform ligament, that is what the examiners wants to know, the falciform ligament, the extent of it. You see that this particular falciform ligament goes in here and limits over here. What structural lines over here in the purple line? The diaphragm, the future diaphragm, right? So this particular higher limit of this thing is limited over here, coming from the diaphragm and getting into the liver, splitting the liver anatomically into right lobe, left lobe. In between them it is running and then it is falling on the anterior body wall, and in falling in the anterior body wall ending into this particular structure, we call them as umbilicus. So the higher limit of the falciform ligament is under diaphragm. Anteriorly it is going on the anterior body wall, inferiorly it is limited by the umbilicus. So this falciform ligament itself is a short note. So whenever it is given as a short note, the embryological considerations need to be addressed as well. Clear or not? So this element you have to address related to these things. Next, moving to this next chapter about this particular foregut tube, hindgut tube and midgut tube. So each of this gut tube have got their dedicated blood supply. So they have their own blood supply which supplies the foregut region, midgut region and hindgut region. So they have been separated out. So there are no mixing over there between this particular blood supply is there. The foregut, which is going to be developed in here, supplied by an artery, we call them a celiac trunk. that we are going to dissect into these practical classes. The midgut is supplied through this particular thing, we call them as superior mesenteric artery. The hindgut is supplied from the inferior mesenteric artery. In between these particular arteries, they are having connections between the overlap regions. So, the superior mesenteric artery, inferior mesenteric artery and the celiac trunk, they are dedicated to give the blood supply to this foregut primitive. So, whatever the primitive foregut yields in, going to be supplied by the branches of celiac trunk. Whatever this particular midgut tube yields in the adult, they are going to be supplied by the branches of superior mesenteric artery and likewise the hindgut. That is why it is easy to understand the blood supply of the gut. So, if you remember that where from this particular embryological part of the gut is developed from, the blood supply will be easy to understand. Even if you cannot remember the exact branch, you can say this is supplied by the celiac trunk branches or superior mesenteric artery branches, inferior mesenteric artery branches. So, that is why this demarcation, how far this foregut, how far is the midgut embryologically and this midgut foregut tube has developed, that what you have to understand. For the foregut, celiac trunk, midgut, superior mesenteric, hindgut, inferior mesenteric artery, and these are your MCQ questions. They will put a MCQ question, this particular branch which is going to be supplied like cecum is supplied from a branch of superior mesenteric artery, inferior mesenteric artery or celiac trunk. So, you have to choose the right answer. You have to know that where from the cecum is there coming and belongs to which part of the gut embryologically and supplied accordingly. So, let us take it down the differentiations of the gut tube. So, we will beginning with this particular foregut tube, then we move to the midgut, hindgut later on. So, foregut derivatives. So, foregut tube, the primitive foregut tube that was this tiny small starts to develop, starts to progress and develops into or leads into this. So, from the foregut tube which is present in the cranial fold of this particular developing embryo gives rise to the esophagus, gives rise to the stomach, gives rise to the some organ like liver, gallbladder, bile duct, pancreas and upper part of the duodenum. Now, guess what? All these organs which is developing from the foregut tube derivatives are going to be supplied by which branch of arteries? Celiac trunk. Eyes closed. So, these particular organs and this part of the gut tube are going to be supplied by the branches of celiac trunk. That is what you have to know, where from it is beginning and where does it end. So, the end part which is junction point, where is the junction point? We call them as upper part of the duodenum. So, duodenum anatomically, gross anatomically, we divide into 4 parts, but embryologically we divide into 2 parts, upper part, lower part. So, we will be addressed how far is upper part, how far is lower part, and there is a mix up between that particular region. So, whenever there is a border region, there is a dispute. Likewise, in our life we have seen that. So, anywhere you have put a barrier between this is yours, this is mine, there always have been a barrier dispute. So, here as well, wherever there is a barrier region or this particular dispute region which lies over here, the transforming zone, upper part and lower part transforming zone, we have a dispute region. We will be talking about the clinical problem on that particular segment. So, foregut tube, esophagus, stomach, liver, gallbladder, pancreas and duodenum, and all of them are supplied by the branches of celiac trunk. Celiac trunk is an artery. Now, why do we call them as celiac trunk? Like it looks like a branch of the tree which is sprouting or branching out. So, it has a very small base, but it has a large branches. That is why it looks like a tree which is going to give several branches. That is why you call them as descriptively celiac trunk. Rest of the things we call them as superior mesenteric artery. In short, we call them as SMA, inferior mesenteric artery, IMA, likewise. So, this is how these things are addressed. So, can you take more? Little more? Okay, little more. Esophagus, alright. So, this particular foregut tube, we will talk about the esophagus, little bit about the esophagus and the developmental error, nothing much, because those are the things that you are going to talk about. So, this primitive or cranial most part of the foregut tube yields to a tube, we call them as esophagus, which is present in here. So, what is the catch out here in the development of the esophagus? That is what you have to focus. You have to keep focus for few minutes regard to the esophagus. So, esophagus developed from which primitive gut tube? Foregut tube. Which part of the foregut tube? Cranial most part of the foregut tube. Now what happens, or what remarkable thing happens in this primitive part of Foregut tube is that this particular tube is formed over here. There is no dispute or no controversies out there. What thing develops special out here, within this particular anterior wall of the developing Foregut tube, there appears a projection. We call them as diverticulum. So this particular projection, which is coming from the anterior wall of the developing Foregut tube, which is going to yield the esophagus, they show a bulge out position. We call this bulge out things as a diverticulum. This diverticulum, we call them as respiratory diverticulum. That means that's what is going to yield the respiratory system. Now, this particular respiratory diverticulum, as the time progresses, splits into two things, becomes lung buds like this. But we are going to talk about the development of embryological segment, right, of GI system. We are not talking about the respiratory system development. So we take this particular thing, considering that this is developing here and the respiratory diverticulum going to form the future respiratory system, lung bud, etc., etc. We know that, but we are not going to discuss about that. We will be coming back to this particular primitive Foregut tube, which is going to talk about related to the formation of the esophagus, and we go back to that particular region. So this primitive Foregut tube develops into esophagus, and the anterior wall of the esophagus creates a diverticulum. We call them as respiratory diverticulum. So this respiratory diverticulum bulges out and creates a cleft in between them. So this particular cleft, as you see there, it has a highest end attachment, and the lower part of this particular tube or Foregut tube with this respiratory diverticulum is separated out by the mesenchymal tissue, which is going in. So we call them a splenopleural mesoderm. So that particular splenopleural mesoderm slowly getting in between this particular respiratory diverticulum and this esophagus and separating thing out, except the junction point. So this particular separating part which is going on in this particular region, we call them as tracheoesophageal septum. Between the trachea and the esophagus, they are separating out. Who is separating out? The mesenchymal tissue. What mesenchymal tissue? We call them as splenopleural mesenchymal tissue. That's it. So this is creating a breeze between the lung system and the esophagus system, except it has a communication in here. We call them as laryngeal inlet, where it is communicated, which is hanging from there. Rest part is separated out by this particular tracheoesophageal septum, or this particular septum, as you see cranially from above, going to great groove in and separate this particular respiratory tube and separating out with the esophagus, so that the trachea remains separate and the esophagus remains separate. This is how these things are there. Now, there is no rocket science about it. So what's the big deal about it, having this particular separated out? Why we are buying time out of here? Thing is that if by any means this splenopleural mesoderm fails to invaginate, fails to groove in between this particular developing respiratory tube and this growing endodermal tube, Foregut tube, we have a connection remains there. Now what happens if we have a connection remains there? That creates a gap or communication. We call them as fistula, where the trachea is communicated with the esophagus. Now imagine we are born with a communication with an esophagus communicated with the respiratory trachea. What happens to us? The moment we suck in some fluid, the moment we take some kind of element which is food, then maybe going to the esophagus to the stomach, but as well there is an equal 50% chance they are going to land up in the respiratory system. Then it's going to be on our lungs. There's going to be a mess. So it will going to have a choking thing. So that's what happens. So we have a fistula, faulty development of this particular splenopleural mesoderm. We have a big problem. So that particular clinical case, we call them as tracheoesophageal fistula, TEF. TEF is a kind of deadly thing that happens, which is a communication between the trachea and the esophagus, which are trading a free passage for the passage of the food material from this esophagus to the lung system. So the moment the kid is swallowing a fluid, is choking. How the patient is going to come to you with a clinical problem like this? Just born, newborn, coming with the moment it suckles or moment it sucks, takes the milk, it cups out, becomes blue, looks like something is going really bad, cannot take any food anymore. The mother is coming with a complaint like this. Now what are you going to do? আপনি বসুন আমি আসছি বড় ডাক্তার খুঁজে আনছি This is what you are going to do. This is your choice. So this is what it happens if you have a partition gap between them, between the trachea and esophagus, where this splanchnic mesoderm is failing to fall between them and create a partition, and that particular partition is never there, and there is a free trade passage between this trachea and esophagus when you have a problem or hell of a problem. That is what is the most grave thing that happens between the developmental error on esophagus. Apart from that, there are a number of problems. The esophagus may be short. We call them as atrasia, failure to develop proper esophageal length. So esophagus is short, it is pulling out the lung, pulling out the stomach up. So that can happen. So lung may be just like a herniating into this particular thoracic system or thoracic channels because of the short esophagus. We call them as herniations, that particular sliding hernia or any kind of hiatal hernias, etc., etc. The esophagus may not be canalized. The esophagus remains solid. It's never been formed with the tube lumen. We call them as stenosis. It may have other problems. What other problem? The esophagus, the lower end of esophagus, which is going to end into this particular stomach, should have a network of the cells which is helping this particular migrations of the peristalsis, which is done by the cells we call them as ganglionic cells, which is a part of enteric nervous system. We learned that enteric nervous system in the physiology. In like we have 3 sets of systems or 4 sets of system: peripheral system, central nervous system, autonomic system, and enteric system. So this enteric nervous system, which is present in the gut system, which is running this peristalsis and anti-peristalsis for us, is run by this particular cells. We call them as ganglion cells. The ganglion cells comes from elsewhere, migrates in, sets in, and creates this particular migratory piece. So they are lying into a network of plexus. We call them as Orbach's plexus, Meniere's plexus. If you haven't learned in physiology yet, if you miss that in the GI system development, read that because those are your MCQ questions. So this particular migration or failure to migrations of this ganglion cell into this particular networks of Orbach's plexus results in the improper peristalsis leading to the dilatations of the lower end of the esophagus. We call then this esophagus takes a shape of a heart and it looks like a heart in a skygram. We call them as cardiospasm or achalasia cardia. That means it looks like a heart. So it looks like 2 hearts the person is born with. One with a real pumping heart and another is pseudo dummy look like heart. So that's why you call them as achalasia cardia because of this particular ganglion cell haven't migrated. The esophagus where the stomach is joining with, that point is really tight, not allowing the peristalsis to move through. That can happen as well. So out of this 4 elements that we have to talk with, this is what we have to emphasize on understanding here is the TEF, tracheoesophageal fistula. Why this happens, how this happens, what happens to the patient if he have this particular fistula, how they are going to come to you with the clinical problems likewise, and you have to either run through or make a call. This is likely this one and I am going to run through some tests to confirm it. And TEF can be diagnosed in utero. Like still the baby is inside the mother womb, with test we can diagnose it if you have the right knowledge. And for that again you have to run and find a good doctor or deal with your own. So this is what we are ending with, right? You want to more? No more? All right.